Oh, so we are going to continue from where we stopped. So this is going to be the second part of our genetics. So we stopped at number 18, that's monohybrid inheritance. So, and I think I've done justice to that already. But I'll just quickly give a recap of what we've done so far. Okay, yes, okay. So, the crosses we have here, um, like I told you, which I've explained earlier, in F1 generation, there will always be a cross between pure breeds, meaning if it is what's it called? If it is a character for tallness, if it is I'm sorry, for height rather, for height, for color, and so on and so forth. And when you talk about height, for example, so it's either you are tall or you are short. You want to talk about color now, it's either you are white or you are red. So that's based on a plant which Mendel used when he was doing his experiment. So now, but for this very sample that I have here, I actually make use of tall and short. And when you talk about tall and short, a character, a character is what is height. To pure breed, like I told you, the genotype will be pure, two capital T, two small t. Now you come down to the gametes level, meaning this is a somatic cell, for example. So during the production of gametes, that's during meiosis. So that's this what it means here. During meiosis, it forms gametes. So you have capital T, small t, small t. And I think I've explained this cross earlier. So one goes to one goes to two goes to one, two goes to two. So in the F1 generation, you're going to have all heterozygous condition. So the genotype will be what? Heterozygous. So the phenotype, which is the physical appearance, is going to be all plants, all plants. But it is not homozygous tall, it's heterozygous tall. As you can see, one allele is for tallness, the other allele is for shortness. So it is heterozygous tall. Now, in the F2 generation, I said F2 generation is gotten from the cross between two offsprings from F1 generation. So we pick two offspring from F1 generation and we move it in here, which are the genotype 1 and 2. At the gamete formation, the genotype will divide. So we have capital T small t, capital T small t. And during the crosses, capital T goes to capital T goes to small t goes to capital T, small t goes to small t. And at the end of F2 generation, you will notice that genotypically, genotype, that is the genetic constituent. The genotype is two capital T homozygous or two heterozygous or we have one homozygous or rather, two heterozygous or and one homozygous short genotypically. Now for phenotype now, it will be one because this one, two, three. The three of them are going to be tall. To be assumed that they will all be tall. So this will be the only short offspring. But like I told you, those small t, they are what? They are recessive gene. They will not express themselves now. But when there is a probability of another cross coming up again, then they can begin to express themselves, just like what happened in F2 here, where you now have a short offspring. So, in summary, That is what monohybrid inheritance is all about. Like I've earlier told you, it's Mendel's first law of inheritance, and it actually talks about the inheritance of one pair of contrasting character. So we are only talking about one character out of all the characteristics that an organism has. Remember, character is also equivalent to trait. So you can see inheritance of one trait. If it's going to be the trait for height, the trait for color, the trait for hair shape, and so on and so forth. So monohybrids deal with just one character. So when you talk about dihybrid now, that means two character or two traits considered. So this is just one now. So we'll get to the hybrid inheritance. So this is 19 dihybrid inheritance now. For dihybrid inheritance, Mendel's second law of inheritance. It is the inheritance of two pairs. Two pairs. Di means two. Mono means one. Two pairs of contrasting character, e.g., a pure breeding round and yellow seed plant is crossed with a pure breeding wrinkle and green seed plant. Now, what are we trying to talk about here? We are talking about shape. Shape. It is either it is round or it is wrinkle. That's a shape, round or wrinkle. And color. It is either it is yellow or it is what? Green. So, like I told you, we are considering two characteristics in an organism. Now, here now we are looking at shape and color. They notice that in this very particular plant, which is pea plant, that Mendel consider, he noticed that when what's it called? When they have a round shape, the seeds are always kind of yellow. And when they have a wrinkle shape, the seed is always kind of green. I get him. Now, it doesn't mean that it will always go that way, but that is assumed to be what's it called? That is assumed to be um the pure breed. So when you have the pure breed, that is what the pure breed has. But in the process of what's it called? Meiosis, during meiosis, the first phase, um the prophase, during this very stage, then you will have crossing over and then things are going to change. And that is what we will be explaining fully. It's a bit long process, which I'll still explain everything. But I just want you to have the concept and understand what we are going to end up with here first. So here now, 2 capital R, remember, R stands for what? Round. So, and if you are talking about a round shape, what it means automatically is it is going to have hybrid. Sorry, it's going to have homozygous round, 2 capital R, because it is a pure breed. 2 capital Y is a pure breed. Y for yellow, R for round. And for the other one, small letter R, so the small letter R will stand for the wrinkle. Small letter R will stand for the wrinkle, and the small letter Y will stand for the grain. So at the end of F1 generation, we are going to have the mixture. There will be mixture there. I'll still explain like I've earlier told you. So I'll still explain it so you'll be able to understand the concept better. So capital R, small r, capital Y, small y. So at the end of F1 generation, all round yellow seed plants is what we are going to see. Capital R, small r, capital Y, small y. Round yellow, round, round yellow. Remember, like I told you, this is heterozygous round now. Heterozygous round. And this is heterozygous yellow, not homozygous again. After the crossing, you have the F1. Now at F2, now remember, F1 is always a cross between two offspring from F1 generation. This is all what this is going to give. It's going to give four of this. Four of this. I still have the cross at the other part of the slide, so when we get there, I will explain fully. But generally, just have it at the back of your mind for that at the end of F2 generation in dihybrid inheritance, the phenotype you'll be seeing is 9331. 9 round yellow, 3 round green, 3 wrinkle yellow, and 1 wrinkle green. That's going to be the results of the hybrid inheritance. So I'm going to explain that process by process. We have it down after the whole terms we are considering here. So I have everything down. So we'll just go from there. So let's go to hybrid itself. What is hybrid? Offsprings of unlike parents. Hybrid. Offsprings of unlike parents, e.g. Mendel's P or Garden P or Drosophila. This leads to variation. It is hybrid vigor, which is also called heterosis. It refers to healthiness of organisms resulting from crossing of two unrelated, less vigorous parents. So basically, what we are trying to do in hybrid is we are trying to take two different organisms that are not, that they don't really look alike. They are not from the same parents. We are trying to cross them so that we'll be able to retain some characteristics. To be able to retain some characteristics which can be passed from generation to another. So hybridization now is used to improve plant and livestock production. So like I've earlier said, you are trying to preserve some character in your animals. Maybe you notice that there's a particular animal that is that grows very well and become very big. For example, when you are into poultry, so you notice some varieties are big, but they are not too strong. But the other ones are very strong, but they are not big. So you can decide to begin to mate them and produce new varieties or new hybrids that will be able to last long, and at the same time, they will be bigger than the parent. So that's the word hybrid there. So we have diploid here. It's the number of chromosomes in the body or somatic cell. I think I've talked about this many times. Even while we're talking about cell, when we're talking about mitosis and meiosis, I've repeated it times with that number. So that is for the diploid. And haploid too. I remember talking about haploid too many times. Anytime we talk about haploid, we are referring back to gamete. So it's the number of chromosomes in the gamete of an organism. And it is represented by the symbol N, so which means 23 number of chromosomes. And the gametes at certain stage in the life cycle of plants are haploid. So that's at the gametic stage. So the gametes, every gamete, the sperm cell, the egg cell, we always have haploid number of chromosomes. In man, for example, now it is always what 23 number of chromosomes. And the next one is 23, aneuploidy, is half the daughter cell produce, half the daughter cell produce as an extra chromosome. Okay, just give me some few minutes to kind of relocate. So sorry for that. I just I'm doing this at night, so I think the family are already in bed sleeping. Sorry for that. So, like I told you, I said it's going to be more of um raw class. So I'm not doing any editing. I said, so let's have it. I want you to be laughing there, so just be laughing. Don't no worry. So let's go to it here. I just want to make sure we're able to cover up almost all the topics here. And for your exam, you have everything, so you can always refer to it, watch it anytime if you want to. If you watch, and it should be okay. Like I said, 23 aneuploidy. Now we've talked about diploid, we've talked about haploid. Now there are some cases, they are not really normal cases actually, and those are the aneuploidy and polyploidy. They are not really normal, they are kind of some kind of abnormality. Let me put it that way. Um, Remember in meiosis, we um, talk about what's it called, sister chromatids, non-sister chromatids, dysfunction, um, chest matter, crossing over, and all those stuffs. Now, during this process, there are times when things are supposed to cross, and after crossing over, in which they are supposed to separate back after exchanging genetic material. Yeah, periods where they're supposed to exchange separate back, and everything goes back to normal after they've exchanged. But in some cases, they will not they will not divide. Like the parts, the edge of one chromosome and cut off and attach itself to the other one. And so these cases, as I said, it's not really a normal case, so but it happens. And that's what is leading us to this aneuploidy. Now, as the daughter cell produce as an extra chromosome. Now, the daughter cell that is being produced, which is supposed to be in during gamete formation, and which is supposed to be N, I get them, which is supposed to be N, just N, which is 23. And if it is the body cell, which is all you're supposed to be 2N, which is just 46. Now, half this daughter cell produced now has an extra chromosome. So an extra chromosome is added to it. So it will now be N plus 1 instead of ordinary N for sex cell, and for somatic cell, it will now be 2N plus 1. So why the other half has a chromosome missing? Now, we talk about what's it called? Homologous chromosome and everything during meiosis, anaphase, the anaphase 1 and all those stuff. Now, when they are now supposed to separate, they did not separate. So one from one of the chromosome now cut off and attach itself to the other part, adding an extra to the other part. Why where it's cut off will be negative. That's -1. And that's what we have here. Now, have the daughter cell produced as an extra chromosome plus 1. One half we have extra chromosome. Why the other one we have the other half has a chromosome missing. So instead of N now, it will now be N minus 1 or 2N minus 1. It arises from the failure of a pair of homologous chromosomes to separate during anaphase 1 of meiosis. When they decide not to separate during anaphase 1 of meiosis, so you can quickly go back to our meiosis and go and read what happened from prophase 1 of meiosis to anaphase 1 of meiosis. So when homologous chromosome now fail, when there's a failure of a pair of homologous chromosome to separate, then you will have aneuploidy. So the reason why I said you should just go into meiosis to go and check this out again, it will make it stick. Like I told you, IGM will always ask you to write on them. And these are ways you'll be able to remember when you even see the terms. Whether you like it or not, there will always be 5 questions. Write short note on the following: aneuploidy, polyploidy, this, that, that. Are you getting me? And once it comes like that, you don't have choice. You have to write it. So, like I've always said, look for higher education, 10 in 1, 20 in 1, or whatever, and begin to answer. Go to the past question. As I'm taking this topic, go to the past question and start answering your question the way you want to answer it. Go and look for your past questions and begin to look at them. Once we are done with this 46 terms in what's it called? All these 46 terms, once we are done with it, now go to your past question and see the ones they always ask questions on, so that you can write it the way you will answer it during the examination period. And I've always said that this thing it's really helped me during my own time, because I work with past questions so much that when I saw question in examination, I wasn't bothered, because a lot of them I've answered it already the way I want to answer it. So all I just do is just go in there and pick the ones I feel I'm okay with. And that's just it. So that is, both sets of chromosomes pass to the same pole of the cell. Now, when there's movement to the pole, remember at metaphase, they all come towards the equator. And from metaphase now to anaphase, where they now need to move to the opposite pole. At metaphase, they are the equator, rather. So from the equator now, there will be spindle fiber being formed, and they'll begin to move towards opposite pole. Now, while they were moving towards the opposite pole, then some homologous chromosomes decide not to separate. So both sets of chromosomes will now pass to the same pole of the cell. Now, when it passes to the same pole of the cell, what does that mean? It means at telophase now, when they now divide totally, you now have two cells. One cell is +1, the other cell is -1. That is what aneuploidy is all about. Now, polyploid now, species in which there are 3 or more sets of chromosomes, e.g. in Down syndrome. Now, polyploid basically have to do more with sex chromosomes. It has to do more with sex chromosomes. Sex chromosomes are basically diploid, either XX for female or XY for male. But once there is an additional chromosome added, it becomes what? Polyploid. It becomes polyploid. And you can see that species in which there are 3 or more sets of chromosomes, e.g. in Down syndrome. We'll still talk more about the Down syndrome also. Like I told you, they are like defects, things that are not supposed to happen. Are you getting? But when they are defect, or when chromosomes are supposed to divide and they are not dividing, like this homologous chromosome, it causes extra chromosome going to one or to another side, therefore causing the kind of effect. And in most cases, it used to affect the organism being produced. So in Down syndrome now, you have XXX, meaning instead of a female that's supposed to have XX, the female will now have XXX, one extra chromosome being added. In Klinefelter syndrome, that is, they'll be having XXY. So this person will be a kind of female and male, but it's good. This person is a male now. This person will have more. This person will be male rather. He will have more of female character than male character, because the person already has XX, which is a complete female chromosome, and another male again, which is Y. We'll still talk more about that, like I said, but just take note of those terms. Polyploid. When a species has 3 or more set of chromosomes, 3 or more polyploid, 3 or more polyploid. Take note of that. Okay, so we go to 25. Test cross. 25. Test cross. Test cross. Now, most cases, test cross and back cross, they used to go hand in hand. And during IJMB exams, they can say write short note on test cross, they can say write short note on back cross. Or any which way, it's still more like going back and forth for the two of them, which we are going to quickly explain now. Now, test cross is a cross between a genetically unknown individual with a homozygous recessive individual. Now, I want you to just take note of something. This is very simple. And I'm going to explain it here now with these diagrams here. So you'll be able to understand how to define test cross using this diagram. Most of these definitions during my own time too, I don't really know them. But because of those crosses, once I remember the cross for that term, I can always use the cross to explain it. Now, this is it. A test cross. Okay, it's just very simple. Test means it's like you are testing something. You want to know what is going to be the result when you test it. Are you getting me? And in most cases, it's either you test with homozygous parents or you test with heterozygous parents. So that is why we say it is a cross between a genetically unknown individual. Now, the genetically unknown individual means we don't know the genotype of that individual. But the two genotype that is possible is either it is homozygous or it is heterozygous. Those are the two possible ones that we can have. So you are crossing a genetically unknown individual with a homozygous recessive individual. It will always be a homozygous recessive individual. Homozygous recessive. Homozygous recessive. It will always be. To determine, remember, we say it is test. We are testing it. So why are we testing it? To determine whether individual in question is heterozygous or homozygous for certain allele. We want to know if the individual in question is heterozygous or homozygous for certain allele. So that's why I said homozygous or heterozygous for a certain allele. Please take note of that, very important. And it is used as a test for linkage, and it is also called backcross. It is also called backcross. So that's what I said. I said they will ask you write short note on test cross or backcross. Now let's quickly look at this before we go to the backcross down there. Now, genetically unknown individual, like I said, we don't know. What we are trying to say is we don't know who this person is. So in exam, if they want to ask question in exam, this place they will put question mark, meaning they don't know the genotype. The gametes too, they will put question mark, meaning they don't know the gametes. They can now give all these results here. So from this results here, you can get what we have here. Are you getting me? Or they can just give explanation. Genetically unknown species give rise to so this, so that, and at the end of this, you understand. So by the time they begin to tell you genetically unknown, or this is this or none, then you should know they are talking about test cross and back cross. So now, homozygous, when you cross it, all F1 generation will be what? Heterozygous, right? Now, you will now take heterozygous condition now. So is this heterozygous condition, one of it, you will now cross it still with the parents. Are you getting me? So it's more like saying you take pure breed, you first get the F1 generation, you now take one offspring from the pure breed, you now cross it with the mother. That's how the explanation looks like. You now take one child and cross it with the mother. Yeah. That's almost what it looks like. But the explanation is genetically unknown individual. You are crossing it with what? Homozygous recessive. Now, for the heterozygous stage, so you now pick the heterozygous genotype, you cross it with this homozygous recessive individual, and at gamete formation, gamete 1, gamete 2, gamete 3, gamete 4, when you cross it, you notice that you have what? Heterozygous, two heterozygous condition, and two heterozygous tall, and what? Two homozygous short. So that is always the result in a kind of in this test cross, because you are crossing a genetically unknown individual with homozygous recessive. So back cross is a cross between the heterozygous. Now in back cross is more of this second diagram. Because test means you are testing it. So because you are testing it, you'll be using the homozygous and the heterozygous. Now for back cross now, it means you are starting from the back to the front. That's what it means here now. So it is a cross between heterozygote offspring. Remember I told you heterozygous offspring and homozygous parent carrying recessive gene. Homozygous parent carrying recessive gene. So for back crossing, it's more like you are taking a child and crossing it with the mother. So that is crossing, back crossing. So it is an application of Mendel's second law of independent assortment. Like I said, we talked about the hybrid inheritance. The hybrid inheritance deals with Mendel's second law of independent assortment. We'll still talk about it after all these terms we are dealing with. So the ratio of the offspring will depend on the original unknown parent, that is PT or capital T and small p. The ratio will depend on the original parent. Like I said, just remember, first cross is you are crossing genetically unknown individual with homozygous recessive. But for back crossing now, crossing, you are taking homozygous offspring and crossing it with what? Homozygous parent that is carrying a recessive gene. That is test cross. Thank you very much. Back cross rather. So go to 26. What is a leader gene? A leader gene is a gene that has no effect on phenotype unless when it is homozygous. When a leader gene is homozygous, it has effect on the phenotype. Normally, it is called a killer gene. It's normally the gene kills the individual bearing it. So another name for leader gene is what? A killer gene. So if you see write short note on a killer gene or a leader gene, so that is the same thing. It has no effect on the phenotype, the physical appearance. You don't see it unless it is homozygous. So if it is homozygous, if there are two, and two and of the same, that's homozygous. Remember that it has effect on the phenotype, and it's normally kills the individual that bears it. It will kill that individual bearing it. So we call it a leader gene. So mutation, we'll still talk more about that when we get to sickle cells and all those stuff. So we'll talk more about that. Mutation is stable mutation. It is a stable inherited change in the gene. It is stable and it is inherited in a gene. That's at gene change or point change or chromosomal change, sugar change, and all those different parts in which changes can occur on the chromosome. So I said it is a stable inherited change. It is also called mutagenesis. It is either induced or spontaneous. Now, okay, let me just read everything here. So I will now explain it fully so you'll be able to understand what mutation is all about. But I know you must have heard about mutation before. So I said it is also called mutagenesis. It is either induced or spontaneous. It is one of the important source of variation among living organisms, e.g. sickle cell anemia. Mutation is caused by radiation, environmental condition, chemicals, etc. Now, when there is a chromosomal change, a lot of things happened. And that is why, okay, let me just put it this way. There's this study by one of these albino presidents. So he said when you give birth to an albino, and remember albino don't have um what's it called? Wow, I skipped my mind just now. Um Black pigment, the black pigmentation that we have in our body, melanin, they don't have it. They don't have melanin. But their president says if you give birth to albino and you don't want the child to be an albino, expose the child at very early stage. Expose the child to early morning sun, the one that give vitamin D, not 1 o'clock sun, not 12 o'clock sun. Early morning, like you wake up in the morning, that's I think between 9 and 10, 11. Yeah, those early morning sun. Expose them to early morning sun. So early morning sun has a kind of vitamin D, and because of the radiation, it tends to change the chromosomal arrangement. Are you getting me? And kind of induce melanin into the albino child. So as the albino child is growing, instead of the child to look totally like the normal albino that we all know, you will now notice that they will now change. Like they will not look like that albino you are expecting. They will not look like every other albino. They will still have their own skin will still be different from albino. So that's an example of mutation. Are you getting me? The environment, um, whether it is being induced, whether it is spontaneous, and also that's for environmental condition, for example, chemicals. Now, if you've watched all this Fantastic Four, Spider-Man, different this thing, those are examples. Radiation or chemicals. You can see they went to the moon or they are in the space, and there was this heavy light that falls on them, and the next thing, the one begin to stretch, the other one looks like rocks, the other one looks like this. Aha, those they are trying to explain what mutation is all about, because that very um that very chemical or that very radiation changes the chromosomal arrangement. Are you getting me? And that is why we say it can be a stable inherited change, because it will remain with them throughout. It will remain with them because the chromosomal arrangement has been changed, has been induced. So even if scientists want to work on any individual they go to the gene, they go to the chromosome and begin to effect, put some things into it so as to um, change the chromosomal arrangement. So this is what we call mutation. Now, what are mutagenic agents? So we now want to look at some of the agents. I think I've mentioned them, but also we want to still talk about them. Mutagenic agents. These are radiations, ionizing radiations or particles, X-ray. Now you will see during pregnancy, they will tell pregnant women not to expose themselves too much to X-rays. Are you getting me? Because as you continue exposing the child to X-ray, what the X-ray is doing is fine. You are seeing your child inside your stomach, but you are continuously exposing the child to X-ray, and it can change the chromosomal arrangement of the child. Anything can happen. The child can turn into anything due to the radiation. Anything can just happen. Are you getting me? Yeah, to some child it can be a positive change, to some child it can be negative change, and so on and so forth. Are you getting me? Depending on the type of radiation they are being exposed to. So gamma rays, but basically you can't say this radiation is good for the body, this one is not good for the body, basically. Are you getting me? But the most important thing is just expose yourself to even sunlight. You will notice that when sunlight, okay, like here that I am now, sunlight here is different from what we have in Nigeria. Normally we are dark normally. But in this their own here, if you as a black man, Even day too, if you don't use sunscreen, if you walk inside the sun for too long, my brother, my sister, it will look like burn. Like when you have a burn, are you getting me? I think you understand what like I don't even know how to put it. To look as if that place got burnt, maybe like iron. When iron, that's how it especially the face. That's how it will look like. So that is why most cases they prefer our own sun. And even as black people here, most cases you have to put on sunscreen if you don't want it because their own sun is different from our own sun. In fact, our own sun is even still good because we can work in our own sun most cases. Pata, your skin will just be black. Finish. So that's an example of mutagenic agent. Because if you work too much in the sun, you'll be dark, even in Nigeria. So what is it doing? It's affecting the chromosomal arrangement for changing you into something else, which is not your real color, like we used to say. Non-ionizing radiation, e.g., UV light, UV light, ultraviolet light. Those are the ones from the sun, from sunlight. So it is non-ionizing, like the X-ray, gamma ray, electron, neutron, and proton. Radiations are highly penetrating. The ionizing particles may alter the nature of gene on the chromosome, thereby causing mutation or variation. So I think we discussed much on that too. 29, carrier. Who are carrier? An apparently normal heterozygous organism. Now, the organism will be normal. Like, you will not see anything in them. They will be normal. They will look normal. But we call them carrier. It carries a recessive abnormal gene. They are carriers. They are heterozygous. Are you getting me? So we have cases of somebody saying you are what did I want to use as an example? Can I use sickle cell as an example? Yeah, we have people that are carrier of sickle cell anemia, but because they are the recessive, sorry, because they are at heterozygous stage. Remember, so one of the genes is affected, but the other one is okay. That is why they will always say, for example, that AS it's not advisable for AS to marry AS. Because you feel like, I'm okay now, my husband is okay now, and why do you say we should not marry? Yes, it's because of the children. Because at a certain stage, there is probability that the two S that you have and X that the wife has, there's probability that they will come together and produce a child. And that child will be sickle cell. That is when you will now know what they are discussing about. So in heterozygous, carriers are normal because they are heterozygous. Remember, heterozygous means one is okay, the other one is not okay. Heterozygous, they are not the same. Hetero, different. So one is okay, the other one is not okay. One is capital T, the other one is small t, tall and short. But because it is heterozygous, it is the tallness you will see. You will not see the shortness until when you marry someone that is short or someone that is also heterozygous. That's when one of your child will now be short. And that's heterozygous. So the same thing is what we are talking about as a carrier here too. Are you getting me? They are carrying the recessive gene, but they might not even know themselves. For medicals, we show that no, don't marry this person because you are SAS, the other person too is AS. So probability of giving birth to SS is what? It's high. So an apparently normal heterozygous organism which carries a recessive abnormal gene. Those are what the people we call carriers. Codominance. 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 Now, the word co means when we say uh, coordinator, coordinator. Are you getting me? So it's more like you are saying or okay, is the word for coordinator what I'm looking for. When you say like what I'm just trying to say here is that we have two people when it is in, anything that has to do with okay, let me just put it this way, co-dominance. So which means there's a dominance. Are you getting me? But this dominance is not fully uh controlled by one gene. It means the two of them participated. Are you getting me? So the two genes or the two chromosomes involved both participated, not only one participating totally. So the condition in heterozygous, where both members of an allelic pair contribute to the phenotype. That's just what I finished explaining now. So it will be heterozygous. Remember, we have complete dominance. Complete dominance is heterozygous too. We have incomplete dominance is heterozygous too. Now we have co-dominance, which is also what? Heterozygous. So which means we have two different genes coming together now. Two different genes are coming together. Now, it is either there is complete dominance, according to Mendel's first law, it's either there is complete dominance, that is red completely dominating white, or there is incomplete dominance in which red and white come together, mix together, and turn into pink. That is incomplete dominance. And the last one here is co-dominance. Now, instead of changing color totally, it's one part can be red and another part can be what? White. Both of us we what? We will show that we are what? We are here. So that is what it means. So it is a condition in heterozygous where both members of an allelic pair contribute to the phenotype. So they both contributed to the phenotype, to the physical appearance. The second point, a mixture of the phenotypic trait produced in either homozygous condition. So it is a mixture of what the two of them produce when they are at their homozygous condition. So the mixture of those traits is what you'll be seeing in this heterozygous part. It differs from incomplete dominance, in which two or more alleles do not show complete dominance. So I think I explained that of incomplete dominance now, not too long. And for that very one, two or more alleles, the alleles there do not show complete dominance, meaning we both come together and we totally change the color. We mix together and we change color into something else. And the something else we change color to does not look like any one of us when we are in our homozygous condition. It doesn't look like us at all. That is incomplete dominance. So we are trying to differentiate between codominance and incomplete dominance. A condition that is different from Mendel monohybrid inheritance. Remember, Mendel's monohybrid inheritance is what we call complete dominance. Red is completely dominating white. Tallness is completely dominating shortness. That is Mendel's monohybrid condition. And the last point, it occurs in both plant and animal, e.g. the production of blue Andalusian fowl. You can go and read about blue Andalusian fowl to have more understanding about codominance. You can just check it online, or codominance, just read about it and get more information once you have enough time. But just make sure we have 5 points here, just go through them. Like I said again, begin to answer your question the way you want to answer it. If you come across it in the examination, and like I used to say, you can predict questions that will be coming out. Yeah, you can predict it. If it's going to be on genetics, the 5 terms they used to ask, 5 terms, which are some of, out of all these 46, you will just pick 5 and that's to write something on them. Are you getting me? If you go from 1994 or whatever past question you lay hands on, extract them out. First one, second year, third year, and look at where they are reappearing. You will notice that some of them they repeat them after 33 years. Some of them they repeat them after 2 years. Some of them they repeat them after 4 years. So if you sit down and you go through it, you will predict those ones that are likely to come out this year. Yeah, it's what I've done times with that number with my students, times with that number. We've done it and it's really worked and it was working and it will continue to work. Yeah. So I've gone for a lot of marking. So I know what I'm actually saying. It's something that works. But I wouldn't tell you I have the time to sit down and go through the whole past question and predict what's going to come out this year. If I have the time, I'll try to do that. But I wouldn't tell you I have the time. But I have some of my ex-students that are teaching presently. I'll ask them to sit down and see and predict the questions that are likely to come. And so from there we can have ideas of what will come out, and you just solve the questions and know them. You will arrange them in your head so that when you see questions, you are not even afraid of all. So, but all the same, sit down and go through all the things one after the other. It's interesting. It's something that you go through and you understand. Even the way I'm explaining it, I try as much as possible to make it simpler for you so you can quickly absorb it. So that is that. So codominance, we are done with codominance. So the next one is cloning. So I know a lot of you have heard about cloning. So it involves reproducing the exact copy of oneself. The way I am now, I can decide to clone myself. Are you getting me? But it's not something you spend ₦1 on, spend millions of dollars on it because of things that will be put in place to do that. If you've seen a lot of there are movies on clones, I think the Calyx Y. It's a very what's it called, very old season movie. I don't know if some of you have seen it also, but if you have Netflix, I think it's still on Netflix. Alex Y. I wouldn't say you should sit down and start watching that now because it's going to take your time. But what they actually did there is one person replicated clone himself. Clone themselves goes through. It's just like taking a stem cell, which is one of the somatic cell. Now there's a way they use it with egg cell. They extract egg cell. There's a way they use it, and they subject it to a particular temperature. The cell turn to tissue turn to organ to organs, system, and like that, it begins to develop. And within shortest time, it developed into a matured human being. I think US, UK, they didn't want them to clone human being because of issues that might arise, which we don't know yet. But most cases, they noticed that some of the animals they've tried it on, which are basically herbivorous animals, those ones that eat herbs, they noticed that they are more aggressive. The cloned one are aggressive than the real ones. So that is to tell you what the repercussion can be. If I have anger issue, for example, now, you now clone me and produce another person that look like me, have everything, the same thing, the same face, the same everything that I have. That's what the person is going to have. And the person is now more aggressive than I am. Are you getting me? And that, let's say I have anger issue, for example, now that person I have my anger issue times 2 or times 3. You know what it means. So that is actually cloning. They've done it for smaller organisms and it's working. So 32, linkage. So linkage is a kind of deviation from Mendelian's principle. And if you notice in life, when they say this is how you must do something, there will always be an exemption. You understand what I'm trying to say? There will always be what? An exemption. So some of the exemption of Mendelian's principle, which is the first law and the second law, one of it is this linkage that you are looking at. So linkage, even codominance too, that is one of it because it's deviated from Mendelian principle. What Mendel is talking about is that when red and white come together, there will be what? Complete dominance. Are you getting me? So Mendel preached complete dominance. But all this deviation, codominance, for example, now, codominance is telling you there's nothing like complete dominance. Both of us can actually participate. Incomplete dominance also is the deviation from Mendelian principle. Incomplete dominance also is saying, no, both of us can come together and change color into something else. Are you getting me? And now linkage. Linkage also is saying, for linkage, it's actually tackle the second law, Mendelian second law. That's why I said when we get down, we'll talk more about it actually. Are you getting me? So you'll be able to see Mendelian's first law, how it is applicable, one character, the second character, the hybrid inheritance, how you will get your 9331. After you know how to get your 9331, then we will now go to the deviation from Mendelian principle, which this linkage now is going against the 9331 rule. It's telling us that we can have two genes, that's dihybrid inheritance, so which means we can have two characteristics that can go alongside each other on the chromosome. That's at linkage. Remember we talked about something under meiosis. We said there's a part where you have linkage, that is when there'll be exchange of materials and all those stuff. Yes. Some cases, there are two characteristics that will link themselves together on the same gene or on the same locus. Are you getting me? Therefore, those two characteristics will be carried together. Two characteristics will become one. Like what we talk about in two-face children now, for example. I said two things is obvious. Their face will look like that of their father, and they will have the type of ear that their father has. So that is more, to me, I can see that is a type of linkage, meaning the physical look is linked to the ear. Are you getting me? So according to Mendel is actually saying, no, the ear we do its own separately, the skin color we also do it, or the facial look we also do its own separately. So it's going to give 9331 ratio. But linkage is saying, no, there are cases whereby you will have the ear color, or sorry, the ear shape or whatever, and the facial look can be inherited together. For those of us that know MKO Abiola, has once been a president, maybe for a day or whatever, before he was assassinated. The same thing with him too. If you see all his children, they look like him and they have the type of eyeball that he has. That's eyeball and facial look, MKO Abiola. If you look at Two-Face Idibia, facial look and ear. So it is linked together. So because it is linked together now, it is a deviation from Mendelian principle. When we get down, I will still talk more about it. So let's just go through this points here. All genes on the chromosome, on a single chromosome, form a group, form a linkage group. All the genes on the single chromosome, remember, genes are found on chromosome. So when there is a linkage, where there is a linkage between them, so they form a linkage group. This is usually passed into the same gamete. It is all usually passed into the same gamete and are inherited together. Inherited together. So we can liken them to be a kind of a sex-linked. Sex-linked, okay? Genes belonging to the same linkage group usually do not show independent assortment. So when we have genes that are linked together at a particular point on the chromosome, they don't usually show the 9331 that Mendel talked about in dihybrid inheritance. Remember, dihybrid inheritance means inheritance of two characteristics, dihybrid inheritance, two. We are looking at two characters together, dihybrid. So under linkage now, because there is a link between two chromosomes, between two genes on the chromosomes now, they will not go with 93331 because they've linked together. Two now become one. So they now behave like one. So at the end of the day, when you are doing the crossing, it will look like Mendelian force principle. So two characteristics now having the outcome of one character because they've linked together. So varieties of ratios are produced. Two or more genes are linked when phenotypes with gene combinations occur less frequently than the parental genotype. Take note of that. Two or more genes are linked. Two or more genes are linked. So two or more, so it can even be more than two genes that will be linked together. You can have facial look, you can have hair shape, and you can have hair color. It can be three, it can be four, two or more linked together are linked when phenotype, the physical appearance, with gene combination occur less frequently. Gene combination occur less frequently than the parental genotype. That's the genetic constituent. Then you have linkage coming up. That's 32. So we go to 33 here now. Sex linkage. Remember, whether it is sex linkage or gene or linkage itself, everything is still based on linkage. The only difference is, sorry, let me get my charger to charge here. A few minutes, please. Okay, we are back. So, I said, remember when we're talking about linkage, somehow we mentioned sex linkage. Now, if it is sex linkage, now what it's trying to say is there are some linkage that are now linked with sex, meaning it's either you see them on you will see them on sex chromosomes, meaning they will link sex chromosomes. So as sex chromosomes are forming, those characteristics also are found on the sex chromosome. And basically, you don't see them on Y chromosome, you always see them on what? On X chromosome. Because X chromosome is only the chromosome that can carry this character. Y chromosome is more like, we call it an empty chromosome, meaning it doesn't allow anything to attach to itself. And you know, for you to have a male child, it is the man that determines the male, that you are going to have a male child, because the Y chromosome is very important for production of male child. So I might decide to remove my face so that I'll be able to see the whole screen for those of you that need it, but you can hear my voice very well. Okay, so okay, let me just get my face from here for a while. Okay, so sex-linked genes on X chromosome slash genes not on Y chromosome. I just explained that you always see them on X chromosome. You don't see them on Y chromosome because Y chromosome is assumed to be what? Empty. It does not allow anything on it. It leads to crisscross inheritance since genes cannot assort independently. Now, crisscross is a type of all the crosses that we've been doing since, that's crisscross. That's what we call a crisscross. So it will lead to normal crisscross, cross X to X, Y to this and that and that. And they don't assort independently, meaning they don't follow Mendel's rule. Are you getting me? The number of recombination of genes are less than when they obey Mendel's law. They are lesser. Phenotypes unequal distributed between sexes. The physical appearance are unequally distributed between sexes because in female you have more, but in male, once only one of the chromosome, which is the X chromosome, has been affected, that is it for the male. But for female, you can have a female carrier, but you cannot have a male carrier. So recessive genes on sex chromosome, e.g. Drosophila eye color. You've heard about hemophilia before. So I think it's one of the terms that we'll still talk about here. So hemophilia is a type of disease also, in which when an individual have a cut or something, the blood clotting doesn't really occur. Let me put it that way. When you have cuts, you will notice that the cells, the tissues and everything in the body, the proteins and everything, they begin to come and start covering the surface so that healing will start almost immediately. But when an individual has hemophilia, they don't have that clotting factor. I think under transport system, we'll talk about that also more. I don't want to go too deep into it, but just have the idea. Hemophilia or hemophilic patients, these are people that their blood do not clot when they have a cut. So therefore, there might be loss of blood and it can lead to their death if not quickly treated. Color blindness is also an example. It can be found on sex chromosome X. We'll still talk more about all those things too, like I said. So color blindness, the inability to differentiate between what's it called, green and red color. So we'll talk more about that. Color blindness, baldness also. So all these are sex linked, meaning you see them on the sex X chromosome. You cannot see them on sex Y chromosome. So when we get to the crisscross whatever, you will see how that is being done. So what I'm trying to say is for hemophilia, for example, now this X now that you're seeing now, you now have H on it. So meaning the X is carrying hemophilic. So for the Y cannot carry anything. It is empty. It cannot carry anything. But since for female, we have XX. So if one of the X is carrying it, the other one is not carrying it. So it's more like the person is what? A carrier. You cannot know. You cannot know that lady has it at all. You will not know. And for the male, the only X for the male, once it has been affected automatically, you will see that the male is this, the male is that. So like this lady, for example, now who is a carrier. The carrier can see an affected person and begin to abuse this person and see you are this, you have color blindness, this and that. But she did not know that she is what? A carrier, which there's probability of one of our child having it. Stuff like that. So, but that's just by the way, just to explain. So we go to the next one, sex inheritance. Sex inheritance. How do we say we inherit a particular sex for a male or a female? So it's due to the inheritance of O chromosome. It's called X and Y chromosome. So when you're talking about sex inheritance, it is either you have X, O chromosome X, or O chromosome Y. In some animals, male is heterogametic. In some animals, male is heterogametic cells, that is having the genes XY. For example, in human being, we have the XY. Our male has XY. Our female has what? XX. Why female is homogametic, which is XX. But in birds, take note of that. Those are some of the points. The position is reverse. Male is XX and female is what? XY. So reverse is the case for birds. Okay. Now 35. Sex determination in man. How do you determine sex? Female produce only one sort of ovum or egg, which contain X chromosome. Female will always have X chromosome. Male produce two sets of spermatozoa. One contain an X chromosome and the other contain a Y chromosome. So there are two possibilities during fertilization now. A spermatozoa that has X chromosome, once it fertilizes the X chromosome from the female, you will have a female, which is XX. But if the spermatozoa that is carrying chromosome Y fertilizes the X chromosome in the female, which is the ovum, then you are going to have a male child, which is what? XX. So sex of the future human is decided at the moment of conception. So once we are talking about moment of conception, the sex has already been what? Been decided. That is why you can see doctors checking their screen to know if a child is going to be a male or a female, because conception already, you know the sex of the child. So 36, yeah, color blindness. I told you we're still going to talk about it. So color blindness now is an example of a recessive sex-linked character. An example of a recessive sex-linked character. A man needs only one copy of the allele to show the phenotype. I think I've explained that. Just only one. Once the X chromosome has been affected, the man will have it immediately. He can inherit this one copy from a heterozygous mother who herself is not colorblind. Did you see what I said now? I explained it earlier. The mother who does not even know that she has it, that is because she's heterozygous. She has a recessive gene for color blindness, so a child can inherit it from there. As simple as that. For a woman to be colorblind, she must have two copies of the allele. You know, I told you, woman has XX. So for a woman to be totally affected, it means the two X must be affected. That is, to be homozygous, both parents must be colorblind or heterozygous carrier of the allele. So that explains colorblindness. And 37 now is human RH blood factor, human resource factor. This has a long stuff. Even when you want to marry, these are some of those things they will ask you to do, and I'll just quickly explain it here. Human resource factor. It was discovered in 1940. 85% of Americans carried an agglutinogen similar to that previously found in rhesus monkey. The person possessing the factor are said to be rhesus positive. So if you have the factor, you are rhesus positive. Those without it are rhesus negative. If you don't have it, you are rhesus negative. No natural antibodies in the plasma of the rhesus factor. Now, what we are trying to say is this: when you are Rh positive, you don't have natural antibodies. No natural antibodies. And you know the function of antibodies? They fight against foreign bodies. So a rhesus positive person don't have natural antibodies. When a rhesus negative person is transfused with blood from a rhesus positive donor, antibodies Rh are produced in the plasma. Now, remember, I said Rh positive, that's those that have the rhesus factor. They don't have natural antibodies. So, I wish I can use something to explain, but I'll explain with my mouth here. Now, rhesus positive individual, they don't have natural antibodies. What are antibodies again? They are kind of white blood cells, and what they do is they fight against any foreign body, any foreign substances, even if it is another blood that is being introduced. Once it is not like them, they fight against it. So when a rhesus negative person now is transfused with blood from rhesus positive, now a rhesus positive person that doesn't have antibodies now, you now give them blood. You now give their blood to rhesus positive, rhesus negative person. Rhesus negative person has natural antibodies. So immediately the blood is entering, because the blood is not rhesus negative. The blood you are transfusing, blood you are giving to the rhesus negative person, because it is not a rhesus negative blood. Natural antibodies will be produced inside the rhesus negative person as a form of what? Defense. I think you understand that. As a sort of what? Defense. Meaning, no, you don't look like us. Did you understand that? So what are we now trying to say now? It means during blood transfusion, a rhesus positive person can give another rhesus positive person blood, because both of them don't have natural antibodies. So nothing is packing anything. Now, a rhesus positive person will not be able to give a rhesus negative person blood, just like what I explained, because immediately the blood is entering into the rhesus negative individual's body, it is going to produce antibodies that will attack the rhesus positive blood, because they see them as threats. They will attack it and destroy it. So at the end of the day, the person might still die because the blood will not be accepted. Now, a rhesus negative person can give a rhesus positive person blood, because rhesus positive person do not have any antibodies that will fight against the rhesus negative that is coming in. And note, the rhesus positive rhesus negative that is coming in, which have antibodies, there is no how the antibodies will be so much that you will go into a new place and you begin to attack them. No. You understand what I'm trying to say now? So that is it. And also a rhesus negative person can also give a rhesus negative person blood. That is because both of them have natural antibodies. So when the antibodies use each other, they will be happy. So it's just as simple as that. So the same thing also for individuals of our no ill effects in marriage. Yeah, also in marriages too, like I told you, they want to know your rhesus factor, so that husband will not be okay, in marriage, let me just explain how that of marriage works. If the woman is rhesus negative, in which the woman has antibodies, if the woman is rhesus negative and the man is rhesus positive, there will be problem, because every sperm cell coming to fertilize the egg cell, antibodies will be produced, and there will be wahala, because they will feel that sperm cell is a threat to their environment. So in marriage, they will advise that no, you cannot marry each other, so that there won't be complications. I'm not very sure, but I think it's one of the issues of is it fibroids they call it. So it will begin to develop like a child, like a structure. Are you getting me? Just to defend any sperm cell coming in. Are you getting me? So leading to kind of fake, I think they call it fake pregnancy or something. Yeah, those are antibodies when they are not compatible. But I think these days, doctors now have a particular injection. I can't remember the name specifically now, that they give So in which when the sperm cell gets into the woman, it will protect the sperm cell, and the sperm cell will look like rhesus negative. Until fertilization happens, everything occurs, and the child would have grown. And also, if it is not detected on time, like I said, if the husband and the wife later got married, they used to say the firstborn might escape it. Are you getting me? But the secondborn might not escape it, because as the child is growing, the antibodies also will be growing against the child. And that is why in some cases, the antibodies can go and attack the leg of the new child and make the child cripple. Or sometimes they go to delicate organs. Sometimes they can attack the eyes, they can attack the head, they can attack the nose. They will just look for a place and attack and make sure they deform the child or kill the child in the process. So that is why it is advisable you take all these tests, not just see any girl and just impregnate them and cause wahala into their life, because you do not carry out the necessary tests to know if you guys are compatible or not. So that's that about that. 38, albinism. So it's a condition in which the external pigment fail to develop in man. The external pigment is called melanin. So when they don't develop it, we call them albinism or albino, resulting in the person having a light skin, a white hair, and pink eyes. Take note of those characteristics: light skin, white hair, and pink eyes. It is caused by a recessive gene A, small letter A, for representing albinism, and only exerts its effect in the homozygous state. So you will see it expressing itself in homozygous state. In heterozygous state, it can be what? Recessive. Remember that? It can be recessive. But in homozygous state, AA, you have them as full albino. It is an example of a single factor inheritance. 39, crossing over. I know we've mentioned crossing over. We talked about chiasmata in meiosis, and so we'll just talk about it here again. So we'll be familiar with them. As we mentioned them, you become so familiar with all those terms. So crossing over, it occurs as a result of the breakage and recombination of homologous chromosome during chiasmata. Breakage and recombination, exchanging of genetic material, crossing over. Chiasmata, it occurs during meiotic division, that's meiosis. In the course of prophase, if you remember your prophase very well, two homologous chromosomes or chromatids are held at a certain point called the chiasmata. Chromatids may break and rejoin at chiasmata. It allows exchange of genes to occur between homologous chromosomes, a process known as crossing over, which promotes genetic variation and evolution. So I think that's straightforward. I've explained it also earlier. Now that is another confirmation. For 41, inbreeding, the word in, you are breeding within a family. Is reproduction with close relatives, e.g. marriages between first or second cousin. Desirable characters are preserved, e.g. speed and perseverance in race horses. Now you notice that people that do horse racing in United States, in United Kingdom, they do more of this inbreeding. They have some particular horse, or they have particular horses that it has always been generation to generation. They run faster than any other horses, and they've made a lot of money through that. So what they do is inbreeding. When they see that one is growing old, they will make sure they cross maybe child with mother, with cousin. If they see that there's another horse from somewhere else again that runs faster, again, they will cross them. So, but one thing I'm just trying to say is it's inbreeding means it is within close relatives, close family. All you are trying to do is you notice a desirable character and you decide to keep it within, so that characteristics will not be lost. So in plants, inbreeding is used to maintain quality and quantities of fruit yield. Most undesirable or leather characteristics, such as deformities, are controlled by recessive genes. So all we are just trying to say is inbreeding among relatives, and the reason why we do inbreeding basically is because we saw good characteristics in the organism. So we don't want to lose those characteristics. So we decide to what? We decide to keep them within the family. Now, outbreeding now, from the word outbreeding, so I can take my own animal and go and mate it with another animal somewhere else also. So that's one of the example of hybridization. So mating of individuals that are not related, it results to the production of hybrids. So you want to produce different hybrids. So you take one animal, you own this thing, you heard that there is another animal somewhere that produces a lot of this, a lot of that. And I think dog lovers, they do that a lot. Yeah, there's one animal is very this is that. You will go and meet them, and they will have offsprings that will now look like this very one. So something like that. So that is what we call outbreeding. So there will always be production of hybrids there, because new hybrids means new offsprings with cool characteristics, new characteristics that you are looking for. So the progeny or the hybrids are often tougher and more fertile, and having a greater chance of survival than the previous ones. So that's what outbreeding is all about. So outbreeding goes alongside with what? Hybridization or heterocase. 43, genetic engineering. I know you must have heard about genetic engineering. So it is the most powerful techniques available in applied genetics and biotechnology. It involves inserting a new gene from an organism. So all this very one here now is like you are extracting a gene from one organism or from one plant, and you are putting it into another plant. Are you getting me? So as to produce something new, or um something that people want. I think a lot of that has been done on our beans. You will hear oloyin, you will hear milk, you will hear olo, you will hear drum. Yeah, different species. Yeah. So what they do mostly is, okay, this is what people want. They want it to be big, this and that. So they extract that. All this work are the work of genetic engineering. So the gene may be newly synthesized or transferred from another organism. It gives us the power to study and to change the genetic instruction of an organism, including ourselves. Other living organisms can be changed for the benefit of human. So like that of beings that I mentioned, and so on and so forth. Even animals too, they've done a lot of them for animals too. And don't be surprised when you hear the word hair transplants and all those stuff. Yeah, all those are still under genetic engineering, because you are trying to introduce a type of hair into that organism and making sure that the the man or the woman or whatever grows a kind of hair. Are you getting me? And or when people are growing new art, new this, new that, just to ensure that it's functioning properly. So all those are genetic engineering. 44, replication. So what is replication? I think we mentioned replication under mitosis, even under meiosis too. We talk about replication. So replication of DNA occurs during interphase. When we're talking about interphase, talk about synthesis and replication. They replicate more DNAs and all those stuff there. So it is important during cell division in preservation of genetic information. It occurs in DNA, that's in the nucleus. DNA, which is found in the nucleus. DNA is double helix. Each strand is complementary. therefore serve as a template for a new strand / duplication of DNA or chromosomes. So I said we will talk about protein, possibly after genetics. So because I want you to just all these terms, all this stuff we're talking about, so you'll be able to what's it called? Put everything together and have a strong understanding about them. So we've talked about DNA now, which is found in the nucleus, which is linked into the gene. So it is a double strand. We have DNA, we have RNA. They are double-stranded, and they serve as template for new strand. So we talk about ribosomal RNA, messenger RNA, transfer RNA. Yeah, so all those stuff with DNA. So every one of them you will understand when we get to the full part where I'll be explaining more about them. But for 45 here, we quickly talk about transfer RNA. And the reason why we talk about transfer RNA here is we quickly look at we said each strand is complementary, therefore serve as a template for a new strand. So you understand all these templates whatsoever. How messenger RNA is being sent out with genetic information, how what's it called, ribosomal RNA produce proteins, and what's it called, amino acids to as a complementary for the other one, and all those stuffs. So transfer RNA now, it transfer amino acid from the cytoplasm, or we call it cytoplasmic pool, to ribosomes for protein synthesis, to synthesize protein. So transfer RNA shows specificity as to type of amino acid. The transfer depends on type of anticodon. Now there's a code that is being brought into the cytoplasm by the messenger RNA. So when the code gets to the cytoplasm, the ribosomal RNA, which has ribosomes, they become active. Therefore, transfer RNA will now do what? It will show the specific amino acids that is being needed, and it will show the anticodon, anti of the code, the other side of the code from messenger RNA. Are you getting me? Because they are specific nucleotides, they are specific base that are supposed to attach. E.g., adenine we always attach to thiamine, A to C, uracil to cytosine. So when we get there, we'll talk more about it. I don't want us to go deep into those ones now, so that I will not some of you will not be confused about it. But basically, all I'm trying to say is the transfer RNA has the anticode. So it's just like you say code, anticode, other side of the code. So it gives that they bind to codon on the messenger RNA in the ribosomes by means of anticodons. So we'll still talk more about it, but just all these terms that I mentioned now, just read them and understand. So when you get to protein, then you will now understand it with diagrams and everything that I will use to explain it, because that makes it very easy to explain. When you see the diagrams, you quickly understand it. So by the time you come here and read this very replication and transfer, you will now understand it better. So that's 45. And the last one, which is 46, is the DNA, deoxyribonucleic acid. Deoxyribonucleic acid, DNA. So we didn't really talk much about RNA here. So we just talk about transfer RNA. But like I said, under protein, we'll talk about both DNA and RNA, so you'll be able to understand them better. So DNA consists of two polynucleotide chains. It's a very long chain, and they are polynucleotides. Poly, like much, not one, not two, not mono, not di. Poly, very long nucleotide chain. Each chain forms a right-handed helical spiral. So it's a form of a spiral form because there are two. So it's more like they intertwine together. The two chains coil around each other to form a double helix. So that's why we always say DNA is double helix. So the chain run in opposite direction. That's antiparallel. Each chain has a sugar phosphate backbone. There's sugar, there's phosphate. That's the backbone. That's the backbone for the nucleotides there with the base. So they have the sugar, they have the phosphates, and they have the bases, which project at right angles. Hydrogen bonds with the bases of the opposite chain. The genetic material of all living organisms and many viruses, DNA. All living organisms, even virus has DNA. So always within the nucleus and has less oxygen than RNA. So it has lower oxygen than RNA. Like I said, DNA and RNA, we'll talk more about them after genetics. So on this note, we are going to end this. So we've been able to cover the whole 46 terms in two classes. Through the whole 46 terms, I was supposed to see how I can finish it in the fourth class, but it doesn't work that way. But any which way, this is going to be genetics 2. So once you are done with all these terms, go into your past question. Look at all the terms that they've asked, screen them out, then predict, like I said, what is going to come out. So when you've predicted something, write everything out. Now, while you are predicting, you will see some that look like, yes, these ones are coming out. First, work on those ones. Write it the way you think you would love to write it in the examination. Write it down so that every day you'll be going through them, becomes part of you. So the ones that are coming under, like, it may come out, it may not come out. Do them also the way you think you answer them. So you notice that at the end of the day, out of the 20 that you put down, 5 is going to come out from there without stress. And you just write and leave the examination up. So thank you very much. So the next one, which will be genetic 3, will now be diving into the main information, where we'll be doing a lot of crossing, answering some questions and others, so you'll be able to understand more about genetics. Thank you very, very, very much. God bless you. We will meet in the next class, which will be genetics 3. Thank you.